Okay, so let's get started. Hello everyone. Welcome to our today's Zoom seminar. It's really my pleasure to welcome Dr. Zhenwen Wen from Tsinghua University to share her latest work with us today. So Wenwen actually currently is a full professor in Institute for Immunology and School of Basic Medical Sciences in Tsinghua. So, so Vaman actually has already been invited to our platform and then talk about her science a while ago. And then I think she actually really did a remarkable progress on her research program. And that's why I think it's worthwhile for everyone to learn her latest science. So very brief introduction about Wenwen. So Wenwen completed her undergraduate from Tsinghua and then continue her PhD training in genetics and development in medical science in UT Southwestern Medical Center with James Chen. After that, she actually did a quite challenging postdoc training, first in Genentech in Department of Immunology, and then she sort of redirect her research interest and relocate to Rockefeller to work with Lab of Molecular Genetics with Jeffrey Fredman, who's working on neuroscience and then metabolism. So from 2014, she came back to China, started her research program in Tsinghua, and all the way up to full professor last year, as just mentioned, full tenure professor in immunology, Institute for Immunology. So Wenwen's lab actually mainly focus on physiological and pathological machinery, which underlying brain-body connection with an emphasis on the regulation, function, and dysfunction of sympathetic and then sensory nervous. Her lab explored the critical roles of immune and nervous systems in metabolic process, aiming to advance neuroimmune-based strategy for metabolic reprogramming. At the moment, her work actually really dive into the machinery of activation and the suppression of the host innate immune defenses and driving the development of novel therapeutics and protective strategies targeting infection and then inflammation through the host immunity. Most of the work in Wenwen's lab actually largely contribute to our understanding of the neuronal regulatory mechanisms, which can governing the metabolism and then expand insight into the neural underpinnings of the pathological conditions such as obesity, diabetes, infections, and neuropathogenic pain. And then also inspire innovative approaches for developing new therapies for the neuronal activity modulation. So as I just mentioned, Woman's work is really disciplinary and then across different fields to connect the immune system and neuron system. Um, Her work has been earned a lot of awards, which including but not limited outstanding young scientist funds, excellent young scientist funds of the National Natural Science Foundation in China. And also, she won the Outstanding Young Scientist Award for the Qiu Shi Foundation Career Award for Women Scientists from Chinese Society for Cell Biology. So, well, I'm a big fan of Wenwen's work. So, I'm very much looking forward to a talk. Please go ahead. Thank you for coming to our platform.
Thank you. I want to thank Tuan for the extremely nice introduction. It has been really supportive to not just to me and all the community. I think we are all very grateful to have this platform to interchange our ideas on science and also potentially building collaborations based on this communication. So I'm extremely grateful. So today I'll mostly share our research on the sympathetic regulation of metabolic activities. So I'll just briefly overview the sympathetic response and also sympathetic nervous system. So sympathetic response has been largely known as a fight or flight response to stimuli, particular stressors. For instance, in response to life-threatening conditions, the sympathetic nervous system can be quickly engaged. That causes the activation of adrenergic receptors within many effector organs and also signal to the adrenal medulla in the adrenal gland. So that can occur in the time scale of seconds to minutes and mobilize the body to defend the host against the many foreign threats. And in the time scale of hours, we'll see activation of hypothalamic to pituitary axis, for example, including production of ACTH, TSH, JH, FSH, or LH. That will signal to many of the organs to engage the endocrine system. Well, in days to weeks, if that stimulation persists, we'll see alteration, for example, in the inflammation immune system, alteration in cytokines, mediators, behavior, so that will cause a lot of consequences. So that's really part of important aspect of physiology as well as pathophysiological aspects. So in terms of sympathetic nervous system, we know for a long time that that's stand for the fight or flight response. And it's known most of the cases it counteracts with parasympathetic nervous system. So for example, in many of the organs or tissues, we would see the innervation of sympathetic nerves that can, for example, dilate pupil, inhibit saliva production, dilate the bronch in the lung, and also accelerate heartbeat, stimulate epinephrine or norepinephrine release from the adrenal gland, and stimulate glucose production from the liver, inhibit the GI tract, for example, stomach, and also as well as pancreas and intestine functions, and also inhibit many other functions as well. So one of the key function it can be involved is the metabolism. So it can regulate lipid and glucose metabolism. It can alter immunity, as we have seen with many of the recent studies. It can also, of course, cause alteration in the behavior and the emotion. So on the signal transmission level, we know the sympathetic nervous system it mostly descending from the central nervous system, and the neurons receive the stimulation from the brain and then use the preganglionic fiber to extend to the ganglia in the peripheral, and then those ganglia extend the postganglionic fibers into effector organs, including many of the organs we are familiar with, for example, liver, gut, and others. Also, the sympathetic nervous system can extend to the adrenal medulla, and the adrenal medulla can largely produce epinephrine, also called adrenaline, to be secreted into the circulation and then work on the effector organs as well. So this is on the molecular level how it's transmit the signal from the central to the peripheral organs. So on the other side, we know the metabolic capacity can be quite plastic because many of the metabolic organs have to deal with the energy storage and energy demand requirements from the body. And taking the adipose tissue as an example, the adipose tissues are really heterogeneous across different spots. For example, in rodents, as well as the recent evidence show in humans, we have thermogenic adipose tissue, including the brown adipose tissue and the beige adipose tissue. And we also have white adipose tissue to take in charge of the lipid storage. And so in terms of metabolic activities, we know the brown and the beige are in charge of energy expenditure and thermogenesis, while white adipocytes are in charge of the energy storage, leptin production to maintain the energy homeostasis. So based on the early studies, we know the brown adipose tissue, which largely responsible for producing heat, are highly innervated by the sympathetic nervous system. So basically, the brain can control the brown adipose tissue quite tightly because of the sympathetic nervous system. It can release norepinephrine, the neurotransmitter, to the tissue, quickly mobilize the thermogenic ability, then produce heat to maintain body temperature, especially important for rodents. While it was known for a long time how the white adipose tissue can receive the input from the brain, because it was largely known that the white adipose tissue are for energy storage. So the original assumption was it doesn't need the direct innervation from the brain. So a few years ago, we started our study by showing that actually in the white adipose tissue, it was not the case as we assumed at the beginning. Instead of like what we presumed, it might just receive sparse innervation from the sympathetic nervous system using, for example, synapsophysin PDP 9.5 or tyrosine hydroxylase TH. We actually see this Y-pad, we saw really dense innervation of the sympathetic nervous system. And also by quantifying many of the cells, for example, tyrosine hydroxylase labeled nerves and the paralipin labeled white adipocytes, we can see almost more than 90% of the adipocytes are possibly receiving direct input from the sympathetic nervous system. So that led us to speculate that those nerve fibers may play really profound and precise roles on regulating various cell types in the tissue. For instance, one of the cell type we got into at the beginning was the beige adipocytes in the tissue. Those we can see the UCP1 producing cells. They are largely formed after 7 days of for example, post-cold exposure, and they are very colocalized with the nerve fibers. And we can see the nerve fibers are really in proximity to the lipid droplet filled adipocytes. So basically, we can see the synaptic vesicles along the fibers. They can be released whenever the neurons are fired. So in addition, when looking into the human Y-fat, we saw that in the human Y-fat tissue, for example, the epiploic, pelvic, mental, or abdominal wall from the human patients, we can use TUJ1 to label the total nerves or TH to label the sympathetic nerves. We can see they are largely overlapping. So basically, the human adipose tissue also receives sympathetic innervation within the tissue. And also to look into how those nerve fibers are regulated, we noticed that some of the tissue residential cells actually produce nerve growth factors. Particularly, this study, we found the stroma cells produce a larger amount of nerve growth factor called NGF. So we use PRX1 to drive deletion of NGF in the tissue. We can see that after NGF deletion, we really ablate the nerve innervation into the tissue. We use PDP 9.5, we can detect the loss of innervation in the tissue. And if we use tyrosine hydroxylase TH to label the nerves, we can also see really the innervation into the white adipose tissue was lost. So basically, the stroma cells determine the innervation establishment in the tissue, and this actually happened very early in the development. And if we put those animals in cold environment, we actually see those fat pad has defects to generate heat-producing cells. So basically, they have defects for thermogenesis. And so by doing the adipose tissue, both in the mice and in the human, we conclude that basically those white adipose tissue receive dense innervation from the central nervous system. And upon cold exposure, they actually undergo dynamic changes. They increase their density to adapt to the energy demand. And on the other hand, if we put the animals, for example, using the high-fat diet-induced obese and type 2 diabetes model, we can see actually the nerve degenerates. So basically that caused the animal to enter a vicious cycle. The brain lost control of the white fat, and that's also lead to deterioration of the metabolic states in the adipose tissue. So basically, we show that an anatomy, function, plasticity, and establishment of the sympathetic neurons in the white adipose tissue. And of course, in the white adipose tissue, not just sympathetic innervation, we know it has many cell types in the tissue. For instance, it's highly vascularized. If we look at the capillary using GPI-HBP1 or using the CD35 to label all the vascular network, we can say it's really highly vascularized. So we also show that really the sympathetic innervation actually can promote hydrogenesis in the tissue that will facilitate the energy exchange between the tissue and the rest of the body. So basically, the wide adipose tissue is innervated by sympathetic nerves, has a lot of blood vessels, which has crosstalk with the sympathetic nerves. And the wide adipose tissue is also populated by many cell types. Stromal cells, as I showed earlier, it can produce NGF to support nerve growth. It can also actually crosstalk with adipocytes and sympathetic nerves as well. So that study started when we look into how the wide adipose tissue responds to multiple stimuli in the tissue. So basically, we know for a long time that the wide adipocytes express many of the receptors that mediate the response to the stimuli from extracellular stimulation. For instance, norepinephrine represent the stimuli from the sympathetic nervous system, which function through the beta-3 adrenergic receptor to cause the thermogenic process. While the wide adipocytes can receive many stimulation, for example, some of the cytokines or growth factors or many other secreted factors from liver or many others. It can also, for example, signal to thyroid hormone or VEGF, many others as well. So we started by looking into what are the predominant receptors which can be expressed in the adipocytes. So one which paid attention is called OGFR because it's really highly expressed in adipocytes. So based on the single cell study, we can see it may be even higher than the beta-3 adrenergic receptor called ADRB3 here. ADRB3 respond to norepinephrine to mediate the response to the norepinephrine from the sympathetic nervous system. So if you look at the expression level of OGFR among the tissue type, for example, epididymal, inguinal, or broad adipose tissue, we can see indeed it's highly expressed. It can be also easily detected in the human adipocytes as well. And also OGFR actually stands for opioid growth factor receptor. It's an endogenous receptor for the opioid. And if we detect, if we assess other receptors for the opioid, we actually didn't see their expression in the adipocytes. So basically, it's quite specifically expressed in adipocytes. So we did straightforward experiments by deleting OGFR in adipocytes using the adiponectin CRE drive the OGFR deletion. So we can achieve deletion of OGFR in those various adipose tissue. While at either at thermal neutral or room temperature, we didn't see much difference on the body weight or fat mass. So only maybe decrease on the epididymal fat tissue. So when we put into the animals into various parameters, like apparatus to measure their difference, we actually noticed one difference, which is the reduced oxygen consumption during the dark phase. Basically, that means they actually has defects in thermogenesis that falls into what we have been focusing on as well. So we found those animals actually they consume less oxygen, they produce less heat in the dark phase when they are physically active. So this is actually due to their impaired thermogenesis. For example, if we look at their thermogenic fat in the YPAT, we really, and when the animals are put in the colder temperature, they really have reduced the beta cell percentage. They also have reduced the level of those genes which are involved in thermogenic capacity, like DIO2, UCP1, and reduce the expression level, the protein level of UCP1 as well. So they really are not that competent to produce heat. And also, if we put the animals in thermoneutral condition, then challenge the animals with cold environment, they really have a difficulty to deal with the cold stimuli. So they drop their core body temperature much faster. Some of them can even, they couldn't survive in the cold temperature. So they consume less oxygen. They have reduced the level of DIO2, UCP1 levels in the cold environment. So basically, we found the defects is due to lipid utilization. So if we put the animals in the cold temperature, we found with time going on, they actually have higher level of free fatty acid in the serum. So basically, they have defect to consume fatty acid. If we stimulate the adipocytes in vitro by giving them norepinephrine to mimic the sympathetic stimulation, we found there that the lipid droplets are reduced, the size was reduced at a slower pace. So basically, they have a difficulty to utilize lipids. So however, if we challenge the animals with glucose tolerance tests, for example, we give them IPGT, they actually has better potency to consume glucose. So basically, they have improved glucose tolerance. And if we foster the animals to force them to utilize the energy store, they also have reduced blood glucose level, either thermoneutral or room temperature. So basically, those animals, they couldn't use a lipid that well, but they tend to use glucose when they are facing like a cold or starvation. And at the cell level, we also checked the respiration or glycolysis of those cells. We can see that those cells indeed they have reduced either basal respiration or maximal respiration. So basically, their mitochondrial functions are reduced, but their glycolysis is enhanced to a certain level. So basically, those cells really they screwed to use more glucose instead of lipids. So those animals, if we mimic the sympathetic stimulation by giving them norepinephrine and measure the oxygen consumption acutely, those animals really, they actually consume less oxygen, they produce less heat. So basically, in response to sympathetic innervation, we think they tend to use glucose instead of lipid for the energy supply. And those animals, though they have improved glucose tolerance during steady state, however, if we put those animals on metabolic stress, for example, giving them high-fat diet, with time going on, they actually tend to develop obesity and type 2 diabetes more easily. So basically, they are more prone to develop those disease conditions. So that now, if we challenge the animals with high-fat diet, they tend to accumulate more fat. So they cannot burn the fat that efficiently anymore. So they have more fat in the adipose tissue also. And also, if we check the glucose tolerance now, after challenging the animals with high-fat diet for several weeks, we can see that now they have actually impaired glucose tolerance and they have impaired insulin sensitivity. So basically, those animals, although they are behaving better in glucose metabolism at the beginning, with the metabolic stress going on, they actually tend to develop the diabetes and type 2 diabetes the more easily. So regarding the molecular mechanism, I don't think we know much. That's so far we can see. So by doing a lot of like co-IP followed by mass spec experiments and localization studies, the only thing we can say is might be correlated with the mitochondrial beta oxidation process. For example, partially in the HeLa cells, we can see the OGFR is partially localized to MTP alpha. This is a mitochondrial localized complex subunit. And the comparison we can make is the MTP alpha is involved in the fatty acid oxidation process. So in terms of the fatty acid oxidation, we know the long-chain fatty acid beta oxidation process, it converted into fatty acyl-CoA, then transported into the mitochondrial matrix, and within the mitochondria, it undergo the beta oxidation process, and it's got broken down by the MTP complex from the long-chain fatty acid to the acetyl-CoA. So basically, this is a long process for the fatty acid to be burned. And there are like a really rare disease conditions called MTP deficiency. In those patients, they have difficulty to convert long-chain fatty acid to energy. So that is a disorder of fatty acid oxidation. So those patients, they actually tend to have really low blood glucose level, for example, after fasting. And they have acetylcarnitine accumulation in blood because those are the substrate which cannot undergo the beta oxidation. So they have substrate accumulation that was spilled into the circulation. So those two parameters, those are what we actually observed with those animals. So they really cannot undergo fatty acid oxidation effectively. They have lower blood glucose level. And we actually did the metabolites measurement, we know that in circulation, they have higher acid carnitine accumulation in blood. So they have the parameters which fit into this criteria. So that's for the OGFR metabolism function. So we know in response to norepinephrine, the adipocytes have to engage pathways to burn fat. So basically, OGFR is among the factors which promote the beta oxidation to facilitate the lipid utilization and facilitate the heat production in this process. And the ligand was, by other studies, they show is met in enkephalin produced by stroma cells. The stroma cells can be one source which produce the ligand and signal to OGFR in adipocytes. So in terms of sympathetic innervation, in the organs, we know not just adipose tissue, many organs receive inputs. For example, adipose tissue receives sympathetic innervation in addition to somatosensory innervation. And the liver also receives sympathetic, parasympathetic, and vagal nerve innervation. And the gut also receives sympathetic, parasympathetic, and also send back somatosensory and vagal sensory back to the brain. And the pancreas also receive parasympathetic, sympathetic, and vagal somatosensory back to the brain. So it's really a complex multi-way communication between the brain and the peripheral. So the next one I will talk about is the study we have done between the brain and the intestine. So basically, the straightforward experiment we did earlier was we checked the adrenergic receptors in the gut. So the mouse has 9 adrenergic receptors. Among those 9 adrenergic receptors, we found that the gut actually expressed ADRA2A in different segments of the gut, like from small intestine to colon. We can see the ADRA2A expression in the tissue. And we also confirmed that indeed it's the epithelial cells which are labeled by APCAM, which are highly expressing the ADRA2A in the tissue. And we can recapitulate the expression of the ADRA2A, for example, using organelle culture as well. So basically, ADRA2A is one predominant receptor expressed in the intestinal epithelial cells. And also in the human, based on the human single cell study, we know the ADRA2A was also highly expressed. Although the human cells tend to express ADRB2, which might represent additional layer of sympathetic regulation in the intestinal tissue. So anyhow, we made a conditional knockout mice by crossing the ADRA2A with William Cree to delete ADRA2A in the intestinal epithelial cells. Now we actually didn't see either IR, oxygen, or heat production in those animals at a steady state. So basically, they didn't show really dramatic defects at a steady state. Also, with time going on, we didn't see much difference on the body weight for the intake at the scale of acute or food intake with a long stage. We also didn't see difference either like fat mass weight or fat mass percentage or blood glucose level at basal state or liver glycogen. We didn't see much difference either. So the difference, the significant difference we saw was the blood glucose tolerance test. We did the oral glucose tolerance test. We actually noticed those animals, they have improved blood glucose tolerance. So basically, those animals can process glucose much more efficiently. And when we do the IPGTT, so basically we inject glucose into the gut, into the peritoneal, we didn't see difference. So that means the glucose has to pass through the lumen to be sensed and then engage the sympathetic regulation to signal to the epithelial cells. At that stage, the DR2A signal is important. And also this glucose tolerance improvement we can see in across many conditions. For example, high carbohydrates condition, high fat condition with 40% of fat and 60% of fat. So we can all see, even with different metabolic stress, this glucose tolerance can be better in the conditional knockout animals in all those conditions. So that with that, we actually turn to the cell types of enteroendocrine cells. because those are the sensory cells in the gut, as many of us, and also Tran has studied that as well. And those cells actually sense the presence of many gut lumen contents, for example, glucose, among many others, that can produce many of the secreted molecules. And the GLP-1 is among one of the key hormones produced by L cells. And GLP-1 has been the drug blockbuster in the past few years because of its role to control blood glucose and reduce body weight. So that's obviously of our choice at the moment, because it's really important for blood control. So when we look at how the sympathetic nerves can regulate L cells, the first thing we looked into is their proximity. So by staining TH to label those nerves, or PDP-19.5 or 2-1 to label the total nerves, we can all see the GLP-1 producing cells. In this case, we label the precursor for GLP-1. We can see certainly many of them are very close to each other. And both like in colon, ileum, we can all see the GLP-1 producing cells are close to those nerve types. And also across the whole mouse intestine, we can see the TH label nerves, or PDP-9.5 label total nerves. They are quite close to the GLP-1 producing cells. So basically, by quantification, more than half of the GLP-1 producing cells may be in a few micrometer distance from the TH positive nerves. And also in the human intestine, we can see similar cases. For example, GLP-1 producing cells, when we co-label them with THPP-9.5 or 2-1, we can see they're quite close to each other, either in small intestine or colon. And also across the whole fragment of the human intestine, we can see TH or PDP-9.5 labeled nerve fibers, or GLP-1 labeled L cells. They can be very close to each other. So even in human gut, we can see in like a 2 micrometer range, we can see more than half of the L cells are actually located within certain nerve fibers. And so when we look at how the sympathetic innervation might affect those L cells, we didn't see much difference on like expression level or total level of GLP-1 precursor in the tissues. But if we look at the GLP-1 production, it's actually a tight regulated process, a signal dependence in presence of luminal glucose. It engage the signaling cascade to engage the GLP-1 release into the tissue. So if we look at the releasing process, we notice that in response to glucose challenge, we saw the GLP-1 actually, we saw less GLP-1 left in the tissue. So basically, that indicates the GLP-1 was released more potently in those conditional knockout mice. So that actually means the model we proposed. So ADR2A signal is a negative regulator of GLP-1 secretion. So when we release the ADR2A signal, we actually have more GLP-1 release. Therefore, we can detect less GLP-1 in the tissue. So basically, it's a negative regulator of GLP-1 production. So when we looked up to see whether the signal from the sympathetic innervation can affect the GLP-1 release, we crossed the THCRE-RT2 animals with HM3DQ to use the chemogenetic activation of sympathetic nerves in the animals. So basically, in this case, if we give the animals CNO to activate the sympathetic nerves and then challenge the animals with glucose, now we have more GLP-1 left in the tissue. And we also tried the viral-mediated sympathetic activation. So basically, we did intracolonic injection of retrograde AAV virus. We injected the THCRE into the HM3DQ animals to drive the HM3DQ expression in the sympathetic neurons, which projected to the gut. Now, if we looked into how the virus can label the sympathetic ganglia, we saw we have a significant level of the viral tracing into the ganglia. We saw citrin expression in the ganglia. we can zoom out some of the neurons which are labeled by the retrograde virus. And in those animals, if we active the sympathetic neurons with CNO, we can indeed see the glucose tolerance was impaired. Basically, when we activate the sympathetic neurons, we have worse glucose tolerance. So now, if we look at the GLP-1 in the gut, we saw that when we activate the sympathetic neurons which protect the gut, we actually have more GLP-1 left in the tissue. So those are chemogenetic activation. If we use genetic deletion now, if we delete norepinephrine production by driving the DBH deletion using THC-ERT2 animals, we can get rid of norepinephrine production in the sympathetic nervous system. Now we can have the animals to be challenged by the glucose. And if you look at the GLP-1 in the tissue, we actually saw less GLP-1 in the tissue. So basically, if we release the sympathetic suppression, we saw more GLP-1 production and secretion from the L cells. And in those animals, if we look at the insulin sensitivity, we actually saw though they can preserve the insulin sensitivity for a long time. That is different from the drug administered situation, because in the GLP-1 drug treated humans, the theory was GLP-1 actually increased the production of insulin from pancreas. Therefore, it can control the glucose level. But in our case, in the endogenous GLP-1, it doesn't seem to be the case. It looks like those animals actually have higher sensitivity to insulin. And in response to glucose, they don't need to produce that much of insulin. They actually have less production of insulin. So that means in the endogenous GLP-1 possibly function through a insulin-independent manner to control the blood glucose level. And also the insulin sensitivity can be preserved even like in the metabolic stressed animals. For example, with high-fat, diet-fat animals, we can still see the insulin sensitivity that is improved in the conditional knockout animals. So for the ADRA2A signal, we actually have been curious if the ADRA2A signal in the L cells is actually not a good sign for the metabolic health, why the animals still keep the signal for a long time during evolution. So therefore, we actually mimic the natural situation by giving the animals limited time to access to the calorie. For example, we only give the animals for 1 hour every day, so they can only take 40% of the calorie into the during that time period time. So now if we check the body weight and the blood glucose, we actually saw those animals really have a difficulty to keep up their body weight. So basically they have a difficulty to maintain their blood glucose level. That will cause problems if they have to deal with the threats. So this is how we reason the conservation of this signaling axis. So basically on the molecular level, we also, with the data I'm not showing here, we also show that the ADRA signal possibly function through as a GPCR-GI couple signaling and inhibit the level of cyclin AMP. Therefore, it can decrease the secretion of GLP-1 from the L cells. So the last question actually we asked if the glucose was reduced in the blood, where it actually go. Because we tested the absorption of the glucose into the system, we think that the absorption might not be the significant contributor to the reduced glucose in the circulation. So we looked into where the glucose can go by using the relabeled glucose scanning by PET-CT. So one region we noticed is the brain, because in the CQ animals, we found that it has faster and more glucose uptake into the brain over time. So we think one possible region the glucose can go from the circulation is the brain regions actually. So we confirmed the glucose utilization in those brain regions. For example, one region we tested is the hippocampus. So we compare the glycolysis process in hippocampus by comparing the wild type and the sickle animals post glucose uptake. So we noticed that those animals actually tend to have a higher level of the the intermediates in the glycolysis process. So basically, those animals seem to utilize glucose more, possibly due to the increased uptake into those regions. And the consequence of this, we can see that from behavior tests. For example, if we check how the glucose utilization in the hippocampus can affect the animal, we put those animals in the Morris water maze to determine their the cognitive function. So basically, we noticed that with time going on, those animals actually spend less distance to platform. They have reduced the latency. They spend more time in the target quadrome. So basically, they can quickly find their targets. One reason being that was they utilize the glucose more efficiently in the brain regions. And so because we started from GLP-1, those animals tend to have more glucose utilization in the brain. So we touched upon little how the GLP-1 signal might affect the glucose utilization in the brain. So one possible contributor is the glucose partition into the brain. So we noticed that when we give the animals exciting food to engage the GLP-1 signal, we actually saw the enhanced oxygen saturation in the brain circulation. So we measure the oxygen saturation we saw in the CQ animals. We actually have slightly increased oxygen saturation. So basically, they seem to exchange more of the energy or substrate with the peripheral. And also in terms of cerebral vasodilation, we also try to quantify the vessel diameters. We saw slightly increased vessel diameters right after we give the animals exceeding food. So basically, that might contribute to infused glucose from the peripheral into the brain regions to supply the brain energy demand. So in this part, I think we found the sympathetic nerve endocrine unit in glucose control. So when we have high sympathetic activity, the gut GLP-1 production can be reduced. That will cause problems in the blood glucose level. So that have increased blood glucose level and reduce the brain glucose uptake. That actually will affect the cognitive abilities. For example, of course, on the counterpart, there are like vagal nerves and other neurons which are involved to counteract the glucose metabolism. So overall, we think the sympathetic activity decrease GLP-1 release and reduce the glucose tolerance. And ADRA2A signal on L cells seems to be a signal pathway which limits the GLP-1 secretion. And the sympathetic L cell unit can reduce glucose utilization in the brain. And this utilization reduction in the brain can hampers the cognitive function. So as a summary of my talk today, so basically I show that the sympathetic nervous system really function as a key bridge between the brain and the peripheral. So basically, either for the adipose tissue or for the intestine, it can regulate key functions of those tissue for like thermogenesis, for endocrine functions, for glucose control. And also, if we look at more comprehensively of the sympathetic nervous system versus the parasympathetic nervous system, we know the sympathetic nervous system can be involved in many more functions we don't understand yet beyond what we currently understanding. For example, in addition to metabolic organs, we know it can regulate like immune organs and reproductive organs and respiratory system, many of them. So I think we still have long way to go to fully understand how the signals can be integrated to really maintain the metabolic health as well as the overall survival benefit to the animals. So with that, I'll stop here. So I would like to thank the students who have done the hard work, our collaborators, and also our founders. So thank you, and I'm happy to take questions and also interact in the future. Thanks.